And this is very important because some people believe that the lower the better. And this is what I said, it is not true. We have to find the optimum level.
We get deuterium from water, air, and food, right? But do we create deuterium in our body as well?
Yes. So when we burn the food from 100 gram fat, we will produce 108 gram deuterium depleted water. And when we burn the carbohydrate, 100 gram carbohydrate, we will synthesize 56 gram deuterium depleted water. But the water coming from the burning fat is not the same the water coming from the carbohydrate. Because when we check the deconcentration of the different food, we found that the carbohydrate is close to the normal deconcentration, 150 ppm, but the fat was 118 ppm, 120 ppm.
Okay, I'm here now with Dr. Gabor again. Thank you very much for your time again for taking some questions about deuterium and deuterium depletion. And we did an interview with some annoying background music before, so this time it will be just nice and clean. And I wanted to start, Gabor, to ask you about deuterium testing, first of all. We are doing a test by people breathing into a glass to collect the breath condensate, and you're testing for us the deuterium level in the breath condensate, right?
Yes.
My question is why is this the way that we test for deuterium, and why would we not test, for example, urine or blood or saliva instead?
We have a chance to test saliva or anything we can test it, but the simplest way is to use pure water, because the equipment we use is a laser equipment, and the laser is dedicated to measure the deconcentration of the pure water. If we use other materials which contains proteins or some other molecules, that would ruin the equipment and that would disturb the whole process. And earlier we measured the deconcentration from the blood. In that case, we use a mass spectrometer. So we have a chance to measure it. The question is, how can we measure it relatively in a simple way and relatively large number of the population? And in that case, the breath and the condensated water looks very simple, can be easily handled, and the measurement should be very accurate. And that this is the reason we try that way. And the most important, this is a non-invasive way to get a sample. So we don't need to cut the people or use a syringe to collect the blood. So this is the reason we recommend to do that.
I see. And is there also a difference in the deuterium level that you would get from a person in the blood, for example, at different times of the day, or what would...
So what we can say that the deuterium level of the water which is condensated from the breath is different comparing to the blood, comparing to the saliva, or comparing to the urine. And there was a paper published by, I guess, by Russian scientists, that in different parts of the body, the D level is not the same because there are differences. And this is due to the differences between the hydrogen deuterium and differences between the molecules, whether it contains hydrogen or deuterium. And this is the reason. On the other hand, when we have a system for one person following the condensated water from the breath and taking the samples month by month, we can see the kinetics, how the deuterium concentration is changing. And at the same time, all the other part of the body will change the deconcentration, but it will keep the differences depending on whether we talk about blood or urine or other parameters.
Mm-hmm. Okay, And one thing I'm interested to find out is about deuterium and its role in different stages of life, from a pregnant woman to an old person. So, like, can you explain like what happens from the beginning, like what role does deuterium play in the childbirth, and what role does it play throughout the course of our lives?
So the first thing what I would summarize, that the original question was, when I started this research, whether the deuterium has any role in the living organism, because the deuterium was discovered in the early 30s, and after that everybody used the heavy water in a high concentration, and they could prove that how big impact on the living organism if they increase the deconcentration. But the deuterium concentration, which is about 1240 millimole in our body, it was simply ignored. So the very first question was whether the deuterium has any role in the living organism. And in the very first experiments, we prepared deuterium depleted water, and we used to, in a tissue culture, and we changed the drinking water of the mice. And the answer was that the lower deconcentration reduced the cell growth, for example, which suggested that the deuterium is necessary for the life, and it may have a very important role in the regulation. On a lifetime, we say that the deuterium is essential optimal concentration in our body, and one of the biggest problems, we say that the population in the last 50, 60 years living their life with a higher deconcentration, just because they are eating much more carbohydrate, and they limited the lipid and fat intake. So the key issue, we have to reduce the average deconcentration of the people with 10, 15 ppm, because we suggest this is the optimum level. And during our lifetimes, the heavy hydrogen and all the heavy isotopes are accumulating in the body. For pregnant women, we do not suggest and recommend to consume deuterium depleted water, because we don't have any kind of experiment proving whether it has any effect. So we simply do not say to consume deuterium depleted water. But on an elder population, we recommend to consume deuterium depleted water, making a cure every year or every second year, and the diet should change to reduce the deconcentration of the body with this 10 and 15 ppm.
I might cut this out of the recording, but what about a child with? I think
we had a couple of child with. In that case, of course, we recommend, and even we had a couple of months old child with
did you do a D test in the breath on the child?
In that time, we didn't test it. No.
Have you ever done a D test on a child with?
Not for the child. No, our main interest is to check the trend and to follow whether there is a progression or there is an improvement.
So would you expect a child with cancer to have a higher in an unhealthy range of deuterium? If you did test
I don't believe that in the child with cancer the problem is the high deconcentration. In that case, there is some trouble in the metabolism, and there is some genetic problem, and this is the reason of the cancer. And of course, we recommend to compensate that type of damaged metabolism of the cell. One way is to repair with lower deconcentration, or to eliminate the cells by reducing the deconcentration.
Okay. So what's your theory on optimal deconcentration for different stages of life? Do you have anything solid evidence, or do you have a theory on what this should be?
Yeah. So the real surprising was when patients came back with a high blood sugar level, and he said that his blood sugar level reduced. And we had a rat experiment at the university, several rat experiment, and at the very first time we made the rats diabetic, and the question was whether the 150 ppm, the normal deconcentration, and the 25 ppm, the lowest deconcentration, there is any difference between the blood sugar level. And it was clear that the 25 ppm reduced the blood sugar level of the rats if they received insulin. If they don't receive insulin, the lower deconcentration didn't count. And then we tested other ppm, 75 ppm, 105 ppm, 125 ppm, and 130, 35, 40, 45. And that was very, very surprising. And maybe it's not a surprising that the best deconcentration was 125 ppm and 140 ppm. So it was clear that not the lowest deconcentration reduced the best way the blood sugar level, but in that range. It means from 150 we went down with 10, 15 ppm. That was the best concentration to reduce the blood sugar level in the rats. And later we ran a phase 2 clinical study with humans, and we published that data, and we could confirm it. So in 30% of the population, the lower deconcentration reduced the insulin resistance, and we reduced the fasting glucose level. Even we reduced the insulin concentration, and that was a positive correlation with the glucose concentration. And the very best thing is that we could explain the mechanism, how it works, because in rat study, we could prove that the lower deconcentration stimulate the insulin signal system way, which means the protein, it is so-called GLUT4, the GLUT4 protein responsible to take up the glucose from the blood. And when the insulin binds to the receptor, that will stimulate the GLUT4 mobilization from the cytosol to the membrane. And this is how finally it's working in everybody, that so finally the lower deconcentration has helped the insulin signal system to work more properly and regulate that whole system on a better way.
So that's your, so because of that, you think there's an optimal range of deuterium, yeah? And well, what evidence is there for if you did deplete your deuterium below this range, what would happen to somebody? Like what sort of side effects could there be for going too much lower deuterium?
So for healthy people, we do not recommend to go below 150 ppm or 130 ppm. But I have heard that in the United States, there was a group, they are distributing water with a 10 ppm. And I heard that those who were consuming 10 ppm for a long period of time, they felt it. That so it means they became drowsy, they had some fatigue, they wasn't so effective, and so somehow it was clear for them that this is not the best way to remove and to dilute all the deuterium. And this is very important because some people believe that the lower the better. And this is what I said, it is not true. We have to find the optimum level.
So we know with that we get water from, we know we get deuterium from water, air, and food, right? But do we create deuterium in our body as well?
Yes. So when we burn the food from 100 gram fat, we will produce 108 gram deuterium depleted water. And when we burn the carbohydrate, 100 gram carbohydrate, we will synthesize 56 gram deuterium depleted water. But the water coming from the burning fat is not the same the water coming from the carbohydrate. Because when we check the deconcentration of the different food, we found that the carbohydrate is close to the normal deconcentration, 150 ppm, but the fat was 118 ppm, 120 ppm. And it means when the mitochondria is burning the carbon source to carbon dioxide and metabolic water, so from fat, we will produce deuterium depleted water, but normal water from the carbohydrate. And this is the reason I guess the big difference is between my grandfather diet, who ate the fat, the bacon, and today the people is recommended to consume and cover the calorie intake 60%, 70% with the carbohydrate. And it means our metabolic water deconcentration is much higher than it was in my grandfather's.
Just for people who don't really understand it, can you explain just simple terms what is the metabolic water and how it is created?
The metabolic water is creating in the mitochondria. So the mitochondria is producing the ATP, and the ATP is a universal energy source for the cell. So when we burn the food, we will get back the energy, but the plant used up the sunlight and using up the energy of the sun, producing carbohydrates and all the other compounds we need. And when we burn it back in the mitochondria, we get back the energy. But so the carbohydrate contains carbon, oxygen, and hydrogen. From the carbon, we will produce carbon dioxide. And from the hydrogen, we will produce water. And this is a metabolic water. And when this hydrogen coming from the carbohydrate, every 150 ppm of the hydrogen will be deuterium. So at the end, we will have a normal deconcentration of the metabolic water. But when we synthesize the water and the hydrogen coming from the carbon change of the fats, that contains only 120 ppm deuterium. So the metabolic water, which is synthesized using up the hydrogen from the lipids, that will be deuterium depleted metabolic water. Every day we produce about 0.3 liter metabolic water, which is not a small quantity. And this is the other reason we do not recommend to people to consume 3-4 liter normal water, because that way we lift up the deconcentration of the body, because we dilute the deuterium depleted metabolic water in our body.
That's interesting. So when you drink, explain to us now, like to deplete deuterium, this takes a long time, weeks or months, yeah, to get to. Now, is that because how much of the water we're drinking is changing the metabolic water? If I drink 1.5 liters of Preventer, how much of this is going to? How many liters of this will change my metabolic?
You are drinking 25 ppm, as I guess.
Yeah.
Or did you dilute it?
No, this is straight 25. Yeah.
25. So this is what I drink. This is 105.
Okay.
Because when you drink 105 ppm, then the equilibrium will be about 130 ppm. And I think this is the best way to keep it, the deconcentration at that level. What was your question?
So my question was, what's the relationship between 1.5 liters that I would drink versus how many liters of metabolic water are we talking about?
So when anybody is starting to consume any deuterium depleted water, that will change the deconcentration in the body. 60% of the body is water. It means people with 70 kilogram, it's 42 liter water they contains. So the very first step the water content of the body will be mixed with the water what we drink, and that will be an equilibrium between the water content of the body and the water we are drinking. And on the other hand, we are producing the metabolic water, and of course, these are mixing in the body, and the volume of these water, which is a part of the body, and the water we are producing will be an equilibrium somewhere. But anybody start to consume deuterium depleted water, day by day we can reducing the deconcentration of the body. The question is where will be the equilibrium, and keeping a stable diet and drinking, for example, deuterium depleted water with 105 ppm, the equilibrium will be about 130, 135 ppm. And you can drink any kind of liter water that will not be lower. If you consume a water with an 85, 65, 45 ppm, then the body deconcentration is going down lower and lower. So in our one of the paper we published a couple of weeks ago, we could show that people consuming water with 25 ppm, 60 ppm, 60 ppm, and closer to the natural, there was a clear linear correlation. The lower was the deconcentration of both water they consume, the lower was the deconcentration of the blood. It's clear. And this is what we can modify, changing the diet, changing the water volume, changing the deconcentration of the volume.
Okay. Well, last time we spoke, we talked about the D level in different foods, and you said about how root vegetables like potatoes will have higher than above the ground vegetables. So can you explain why is that?
Yeah, it is also interesting. So when the different plants fix the carbon dioxide from the air, according to that, there are three types of plants on the earth. It is called C3, C4, and CAM type of plants. Okay. The point is that 85% of the plants belong to the carbon-3 way they fix the carbon dioxide, and this is what typically we eat: the soy, the pea, the wheat, and all these type of food. The C4, which is when someone take popcorn, for example, it has a higher deconcentration comparing with the other plants. And the pineapple even able to enrich the deconcentration of the fruit. So so this is another category, depending what type of food we eat, what way the given plant fix the carbon dioxide, which chemical pathway they fix it, that also can cause some differences of the deconcentration of the different plant. So typically we recommend to eat a plant which use the carbon-3 pathway to fix the carbon dioxide, and not the carbon-4, which is, I would say, the zeomys called.
So carbon-4 plants are root vegetable, potatoes, and things like that. Yeah.
Carbon is, for example, the sugar cane. This is a carbon-4. Typically, which is in the equatorial area of on the Earth.
I've got a question about that, because I was looking at like beehives and snowflakes, and I was looking at the shape is a hexagon, yeah? And so then I realized that's the bond angle between the hydrogen oxygen, right? Is 120 degrees. Is that right? Is that why is that why the snowflakes, although if you look at them, they're different shapes, but they're all based around the hexagon? I think all of them, maybe most of them, but like the then you've got beehives that make a hexagonal shape. So what's this hexagonal shape? Is this to do with hydrogen oxygen bond angle or something else?
So for example, between the water and the heavy water can be some differences depending on this 120 angles, do you say?
Yeah, I'm just wondering, like, what angle does the deuterium make with oxygen and
not the angle is a key issue. The angle, the difference is between the vibrational frequency between the oxygen deuterium and oxygen hydrogen, and this is the source of the isotopic effect. So the size of the deuterium atom is the same as the hydrogen atom, but the vibrational frequency as the deuterium is moving in the OD bond is there are 30% difference comparing to the oxygen hydrogen bond, and it means it takes much more energy to break this bond of the oxygen deuterium bond, and it is true for the nitrogen deuterium and carbon deuterium bond also. So a chemical reaction is slower with a 7 to 10 fold with deuterium comparing with the hydrogen.
Yeah, because I'm wondering why that, going back to sugar and honey, like, is it better to have honey in your tea or cane sugar in your tea? And so I guess honey's better, yeah. And the shape is different as well.
Yeah, I would say the honey maybe has a lower deconcentration than it must be lower than the sugar cane coming from the sugar cane. But they are pretty close to the 150 ppm.
Okay, interesting. So just to go back to one question about drinking you said about the number of liters in a 70 kilo person and how much of this is changing my metabolic water. Do you know, can you tell me in like a rough percentage terms? Because I think this is how quickly do people deplete their deuterium in general, and why does it take that long?
So when we ran the study with diabetic patients, the study lasted for 90 days, but after a couple of weeks, we could see that there is some improvement in the blood sugar level, and they felt better. So on the other hand, when we checked the insulin resistance, I was sure if the study would have lasted for 6 months, we even should have more results. So at least we recommend a 3-4 months consumption of deuterium depleted water to reach the equilibrium and changing all the position, the deuterium for hydrogen. But after a couple of weeks, people feel better because there is a lowering the concentration that can the mitochondria can work more properly, and the whole metabolism is pushed to the right direction.
Okay, and I've got another question about sleeping. Why do you have to be asleep to deplete deuterium? Like, why can't you deplete more deuterium when you're sleeping by breathing out? Yeah, the deuterium. So why is that? Like, why do you have to sleep to deplete deuterium?
I would need more data and more study to conclude something. What I would say that so when no oxygen restriction in the mitochondria, in that case the mitochondria can work properly. When we are doing a loading test and we are running and we are breathing, and the mitochondria do not have enough oxygen to produce metabolic water, then that can disturb this whole metabolism, and we getting acidic, and we need time to adjust back all the parameters. When we are sleeping, then the mitochondria has enough oxygen to couple with the hydrogen and to produce deuterium depleted water. In that case, so when we are sleeping, no oxygen restriction for the mitochondria, and in that case, the mitochondria can work much more properly when we are exercising and we run out of oxygen. And this should be the way that when we are staying, we are calm, we are sleeping, then the produced metabolic water is deuterium depleted. Plus, after a couple of hours, not the carbohydrate will be burned in the mitochondria, but using the ketogens, the lipids, which originally depleted for deuterium, and that can modify the deconcentration of the metabolic water. But we need more study and more data to.
Okay. One other question I had, I wanted to ask you about. Many fruits like bananas and things like that, they cut when the banana is green and it ripens on the container when it crosses the Atlantic. And what's the effect of, like, I know that there's lectins and this kind of thing in the green unripened fruit, but what would that mean also, that the deuterium level is much higher? Because the plant would take the deuterium out of the fruit and into the ground before it's ripe.
The reason the deuterium is higher in the banana, because the rain deuterium concentration is close to 155 ppm. So in the equatorial area, the ocean contains water with 155 ppm, and as the water is evaporating, then it's 155 ppm. In the equatorial area, the rain concentration is higher with 5-10 ppm than in our region in Europe. And the plants will use up that water, the rainwater, and it means that typically the banana, the orange, and all these fruits has a higher deconcentration, just because the surface water has a higher deconcentration. On the other hand, of course, people can eat the banana and orange and all these things. So it doesn't matter in respect, because there are other type of water and there are other food which finally will be an equilibrium again. We do not recommend people to eat fruits coming from the equatorial area, because that way we can keep up on a higher level the deconcentration.
Okay, and so does an unripened fruit have the more D level than a ripe fruit?
What do you mean?
Say an unripe banana, does it have a higher D level than a ripe banana?
I don't know. I don't know any number of whether it has any differences.
Okay, what about for people who live in the region where the high D level in the water and the fruit and everything, where they live? Do they have a different way of dealing with that to us?
I think everybody is adapting to the deconcentration where they live. So it's absolutely normal that people who live in equatorial area, they has a higher deconcentration, and those who are closer to the poles, they have a lower deconcentration. So we believe this is not the key issue. The key issue is whether occasionally we consume deuterium depleted water for a couple of months, which way we can reduce temporarily the deconcentration of the body, and that can repair the body because they eliminate maybe the not healthy cells, the not good cells, the other cells. And the question is whether our metabolism is in a which stage, whether how much we are exercising, what is our body weight, what is the food we are eat, and all together we will have a chance to live our lives with in a healthy position or getting sick and we have to go to the hospital for different reasons.
Mm-hmm. Okay, that's been very, very interesting, and I learn something new every time we talk, so thank you. But
Okay.
Just a final question, can you tell us anything about the latest work you're doing, or any recent publications, or can you tell us about that and where we can find more information?
So our primary aim is to check what is the key mechanism to regulate the cell growth, and what is the reason and what is the big difference between the healthy cells and tumorous cells, and how can we intervene into that signal system. So what we published recently in the control, we checked the pancreatic population, plus we checked the metabolism of the cell lines. And we can say that we could say a threefold increase in the study if the patients receiving conventional therapy plus consuming deuterium depleted water. And everybody knows that one of the common feature of the type is the damaged mitochondria. So in a cancer cells, the mitochondria is not able to produce deuterium depleted metabolic water. And this is a key issue that for healthy cells, and this is the reason I said at the beginning, we need the deuterium, because even for the healthy cells, a higher DH ratio can trigger the cell multiplication. But in the cells, because no properly working mitochondria, the mitochondria not producing deuterium depleted metabolic water, so cells very easily can increase the DH ratio to the thresholds, which trigger the multiplication, and the cell will multiply. This is the reason we published in that paper. The other, it was my very old plan to check whether the deuterium has any influence on the mutation, because earlier it was proved that the heavy water increased the mutation in a virus, and other study also proved that deuterium can influence the mutation rate. And in that study, what we published in the PLOS1, we checked bacteria whether the lower deconcentration can reduce the incidence of the mutation. And the study said that 9 out of 15 experiments significantly reduced the mutation in that system. In 3 studies, we significantly increased the mutation. So the final conclusion is that if there is a deuterium in the DNA, it's not deterministic, which means it doesn't mean it will be a mutation, but the probability of the mutation, the D in the DNA can influence it. So we can say that the lower deconcentration may reduce the mutation rate, but we have to continue our study and to get more evidence that the lower deconcentration can reduce the mutation, which is very important when we talk about a lifespan, 70 years. I checked it, we synthesize about 14 trillion kilometer DNA. So think about, in one single cell contains 1.8 meter length DNA, and our body has 10 to 16 cell division. So we multiply the DNA 10 to 16 times. So it's a tremendous amount of DNA we are synthesizing during our lifetime. And if we can reduce it with a 10% or 5% the mutation rate, it means that no accumulation of the mutation in our body, and that again can help us to stay be healthy for a very long period of time. So that was our last 2 papers, and there is one news. I published my second book only in Hungarian, but I collected all the knowledge what I collected in the last 30 years, and hopefully we can publish it in other languages, which contains all the knowledge, not only me, but...
What is the title of the book, the new book?
The title is Deuterium Depletion, and
okay.
the new approach to cure, to treat, and to stay healthy.
Okay, fantastic. I'm looking forward to the English translation.
Okay, I hope so. You will get it.
I want to just ask one final question and then leave it like that. In one previous video I did with, not with you, but with someone else interviewing that, I said that because it's very early days for hydrogen inhalation, that we don't understand, it's not been extensively studied. And I said on the one video that I'm not sure, I said we might discover that hydrogen might improve the mitochondrial performance of certain cancers. I said that because I didn't know. But I want to ask you something. Do you think there's any possibility that could be the case? And equally, second part of question, can the same be true for deuterium?
So the first thing, when you are using this electrolysis and electrolyzing the water, the inhaled hydrogen is deuterium depleted. So it itself can has an effect because it's deuterium depleted, and it is clear. I guess the hydrogen has a key role in that whole process, and wherever and however we intervene and modified it, it has a high impact and can help us to become healthy and to prevent maybe different diseases and keep the cells in a good position.
So what I'm saying is do you think anything to do with hydrogen, molecular hydrogen, can improve the performance of any cancer? Do you think that's possible? Should be. You think it could increase the performance of cancer? As in be.
Do you mean that the can go faster? Or
what I mean is, can inhaling hydrogen, oxygen, or hydrogen, can that improve the mitochondrial performance of any cancer? Do you think it can?
I guess so, which can be good for the patient taking the hydrogen. No.
Okay, so what I'm asking is, could it be at all negative for the person?
I don't believe so. No, I don't believe so. No,
I mean, I don't think so either, but I just know that it hasn't studied. But in vitro, if you look at what happens to the deuterium and hydrogen, they're working in the opposite directions. Is that right? So the deuterium level in vitro is encouraging the mutations and growth, but the
in vitro the lower D concentration reduce the mutation.
Yeah, that's what we're saying. But what about hydrogen in vitro? What would that reduce as well?
The hydrogen must react with other molecules. So finally, if I think in that case, in this hydrogen story, the question again whether it's hydrogen or deuterium. The hydrogen is very, very reactive molecules, and that will react, and they will produce water or they react with other molecules. And one thing, they can change the deuterium hydrogen ratio, and if they change it and reduce the DH ratio, that can be very helpful and can be very healthy.
Gabor, thank you very much. I'm looking forward to the next. I've got a whole load of new questions coming up soon for the next one. So thank you, and I hope that you produce more research as well, as you're doing is fantastic. We all appreciate that.
Thank you very much. Thank you.
Okay, excellent. Thank you. All right.